Chapter 20 is going to look at DNA tools. So these are the technologies that we use to manipulate DNA. And then biotechnology. So biotechnology is how we take these tools and leverage them for human consumption. There are three main technologies that we're going to talk about in this sequence. DNA sequencing. So this is the first step in DNA technology, is what does the DNA actually say? Restriction enzymes, so these are going to be used to cut DNA, to insert DNA into specific sequences. And then polymerase chain reaction. So restriction enzymes and PCR, polymerase chain reaction, are both used for DNA cloning. So a lot of the times when we work with DNA is such a small molecule that we need to make copies of it if we're trying to work with a specific gene. So we want to make millions and millions of copies of it, and both of those will give us a way to do that. Biotechnology is slightly different. So biotechnology is the manipulation of organisms or their components, such as DNA, to make useful products. So this is talking about making genetically modified organisms and making things that humans are going to buy and consume. So first is DNA sequencing. There have been several implementations of DNA sequencing. The first was by a scientist named Sanger, who had a very slow, tedious method of doing DNA sequencing by looking at individual nucleotides using radioactive labels. In the generations after Sanger, we implemented what's called next generation sequencing techniques, which took the same basic idea but used computers to compile DNA fragments that are copied. So what we can see in the results down here is this is a fragment of DNA, and we use radioactive labeled nucleic acids to pair with these fragments. And we put radioactive labels on these nucleic acids. So whenever a nucleic acid is found, it gets labeled as radioactive. So you can see these results. Anytime the bar reaches up to 1, that's going to indicate 1 nucleic acid, and you can see that that fluoresces is the color T. So this would indicate that there was a thymine here. The next one, again, we're at 1, and it's yellow for C, so that would indicate that this is a cytosine. When there's 2, so you can see that this green bar comes all the way up to 2, that's an A, so that's going to indicate that there's 2 adenines in that spot. So you can see that this one has been translated out for you. There's 2 thymines there. This yellow is a cytosine right there. Then another thymine. The next one here is orange, that's the guanine. And so doing this by hand is extremely tedious. Instead, this fragment of DNA would be fed into a computer, and then the computer would read this results and give you the DNA sequence. And then all of the different fragments that had been used would be compiled in the computer to give you an entire sequence of the entire genome. Next generation sequencing like this is the most common, and it is still used today, especially by scientists who are sequencing large genomes or lots of DNA. It's a very computer heavy process and requires a lot of kind of processing power. Third generation sequencing is kind of the next step. Most often, this is going to be new techniques that are faster, less expensive. These are typically either used in conjunction with next generation sequencing or sometimes replace next generation sequencing altogether. One example of this is the nanopore sequencing. So what this does is it runs a single strand of DNA, so you can see here's a DNA strand, through a tiny, tiny small pore in a membrane. So here's the nanopore. And what happens is as it goes through here, it disrupts the ionic current. And each current change is respective of one of the 4 nucleic acids. So based on what the current says, we can tell whether it's a cytosine, adenine, thymine, or guanine. These are really exciting. These are absolutely tiny. So here you can see the pipette with the DNA, inserting it into the nanopore. So you can run an entire DNA strand on something the size of a thumb drive and then plug it into a laptop and read that sequence even in the field. We have sequenced millions of genomes. So by the time the human genome was done, even a few years later, we had started sequencing many, many other organisms. There's a free catalog online called GenBank. So if you go to this website, you can find free sequences that people have posted to this online resource. Most of the time when we want to study a gene or make something with a gene, we can't do it with a single piece of DNA. A single piece of DNA isn't very much to work with. So a lot of the times we need to do processes that are going to allow us to clone the DNA and make many, many copies. A plasmid is something that we find in a bacterial cell. So this is some kind of prokaryotic bacterial cell. This is the bacterial DNA, so this is the chromosome. And this is a 1 chromosome and it's a circle. You can see it's all contained here. Plasmids are extra small pieces of DNA that are not part of the main bacterial chromosome. So over here you can see this cell has 1, 2, 3 plasmids, and they're just teeny tiny circles of double-stranded DNA. A lot of times plasmids in bacteria are going to convey resistance to antibiotics, but plasmids have proven to be something that DNA researchers can work with. So what a researcher can do is we can extract a plasmid and we can cut it using something called restriction enzymes, and it'll cut in a specific spot. And we can insert a gene into the plasmid. So here you can see they have inserted the gene for insulin. Insulin is the hormone that's going to help diabetics to regulate their blood sugar. And so people who are diabetic either don't produce enough insulin or they have resistance to insulin. So either way, they have to take insulin to help their bodies regulate blood sugar. So what this is going to do is we're going to put the gene for insulin into this plasmid, stick this plasmid back into a bacteria, and then let the bacteria reproduce. If you remember, bacteria reproduce very quickly. So all they have to do is divide, and then you've got 2 bacterial cells, and each one will have a copy of this plasmid. So we can let a bacterial colony reproduce for a day, and we can make millions and millions and millions of copies of this gene inside of those bacterial cells that we can then later harvest. So here's how restriction enzymes work. Restriction enzymes are molecules inside of bacteria that cut DNA in specific sequences. So these are a mechanism for bacteria to use as a defense against viruses. So if a bacteria gets infected by viral DNA, restriction enzymes can often cut up the viral DNA and allow it to prevent it from being infected by that virus. So we have discovered restriction enzymes inside of bacteria. So now what we do is we actually take those enzymes out and we harvest them and use them for ourselves. Bacterial restriction enzymes are going to cut at specific DNA sequences called restriction sites. So this specific enzyme that we're going to use in here is going to cut at the sequence GAA-TTC. And you'll notice that on both strands it says GAA-TTC from 5' to 3'. And so these restriction sites are always going to cut at a specific spot. So this one is going to cut between G and A. It's going to leave this DNA cut into 2 pieces. And those 2 pieces results in restriction fragments. So this is a fragment and this is a fragment. Because of the way that restrictions enzymes cut, they leave a single-ended strand called a sticky end. So right here you can see the sticky end. So the sticky end reads AATT on this side, AATT on this side. So those are the sticky ends. If we then take our insulin gene and we cut it with the same restriction enzyme, it will end up with the same sticky ends, AATT, AATT. So now our restriction fragments and our target gene actually have the same sequences to match back up. So if I put these together into a container, they will align back together. And so we will get fragments that will align with the target gene, and you can see how they match back up. What we need to do then is add DNA ligase. That's the one that's going to bind fragments back together. And here we go. After the ligase, we have double-stranded DNA. It has been cut and our target gene has been inserted. So this is called a recombinant DNA plasmid or DNA molecule. Recombined just means that it's got DNA from 2 different organisms. So now this recombinant DNA, we're going to need to check it. So we check the recombinant plasmid by running it on something called a gel electrophoresis. So a gel electrophoresis is here. There are several wells right here that are places you can insert DNA. And this is a porous gel. DNA has a slightly negative charge because of all of the phosphates in the background. So when we run a power source through it, where we have a negative over here and a positive pole over here, the DNA will move along the gel like this. The larger molecules don't move as far because they're really big and it's kind of moving through a gel. So molecules that are closer to the negative end are going to typically be larger molecules of DNA. The ones that get to move further down here are going to be smaller pieces of DNA. Those are going to be able to move through the gel much faster. And so what we end up seeing is a series of bands of different fragments of DNA that spread out. So when we are making a recombinant plasmid, we know what kind of bands we should be looking for, and we can run the gel and check it to make sure that we actually made the plasmid we were looking for. Gel electrophoresis is also used in all DNA technology. So if a scientist receives a sample of DNA that they're going to use as a study for their research, they check it on a gel first to make sure that it's the correct fragments and size and DNA that they were looking to use. Another way to make copies of DNA, and this is used to make specifically copies of small fragments of a targeted segment. So if we know that there's a specific gene that we want to make copies of, then we can use PCR or polymerase chain reaction to make many, many copies of this very, very quickly. So this goes through a 3-step process. So we start with a bunch of items that we need inside of a test tube. So we have our target sequence, which is the target gene that we want to make a copy of. We're going to have some nucleic acids. So these are nucleotrisophosphates. So those are going to be the adenines, the guanines, the thymines, the cytosines. Those are going to be the new nucleic acids that we're going to be adding. We've got some primers because we know we're going to need primers for polymerase to work. These primers are going to specifically pair with the target sequence that we want to work with. So they're going to specifically target the sequence that we want to copy. And then we have a DNA polymerase. This DNA polymerase is a special DNA polymerase. It was found in bacteria that live in hot sulfur springs. It is called Taq polymerase. It is not the DNA polymerase that's found in humans. This is a heat-stable DNA polymerase. So this is going to be able to function at a very high temperature, unlike the DNA polymerase that you find in our body. So the first step is to open the double helix. So we do this through denaturation. So this is where we do heating. So at this first step, you would heat up the contents to separate all of the DNA strands. What this does is it breaks all of the hydrogen bonds between the two DNA strands, and it's going to allow them to separate. The second step is annealing, so this is cooling. And while it cools, the primers are going to go ahead and bind to their complementary sections. And then the last step is replication. So this is going to allow for that Taq polymerase, so here's my DNA polymerase there, to add the nucleotides into the complementary base pairs. So it'll work from the primer, 5' to 3', just the same as we do in DNA replication, to add these new DNA nucleotides into this. So at the end of the first cycle, we started with 1 double-stranded DNA, and we ended with 2. Every cycle will double the number of copies. So in the second cycle, we start with 2 and we end up with 4. So 2 to the 2 equals 4. By the end of the third cycle, we started with 4 and we end up with 8. 2 to the 3 is equal to 8. So every time we do these cycles, we can get 2 to the n number of copies. So this is going to be very quick. It doesn't take too long to do a single cycle of PCR. In a couple of hours, you can do 40 cycles. So let's say we're on n equals the 40th cycle. So 2 to 40 is a really huge number. It's over 1 trillion copies of DNA. So it's not just double, it's doubling every time, and doubling gets very big very fast. So PCR is a really efficient way of making lots of copies of a very specific small piece of DNA. And this is called DNA amplification, or making lots of copies of.